Hey, I'm Sam. I'm going to give a brief introduction as to what we're going to talk about in this presentation. What we basically want to discuss is the physical properties of viruses and how we can design our own technology based on these organisms. So the first part of our presentation, we will discuss what a virus is, what their life cycle is like, and how they are constructed. This will be done by Isabel. This will then be followed by a brief discussion about the geometry and subsequent geometric restrictions of a virus, and this will be done by myself. Then Alvaro and James will take you through some of the thermodynamic properties and why this is important for the virus to undergo spontaneous self-assembly. Then we will go into a bit more detail about some of the other properties of viruses and why these are important, especially if we want to model our own technology based on these viruses. And this will be done by Ewan. And then finally, Niamh will round up the presentation by discussing some of the ongoing current research into virus-based technology. And with that, I will pass you on to Isabel.
Okay, so I'm Isabel, and I'm just going to be running through the composition of a virus. There's a few things on this slide that I want to bring your attention to. So observations have shown that most viruses have the same basic formulation: a capsid, the viral genome, and in some cases, an envelope. The capsid of a virus is composed of structural protein subunits known as capsomeres. These protein subunits are folded polypeptide chains. The role of the capsid is to protect the viral genome and any other enzymes contained within the viron, which may be necessary for replicating the virus. So there's a few different types of viruses, and they can be categorized by a few factors. These, so I'm going to start with the symmetry, and this is what basically the polypeptide chains that form the capsid can be folded into. And then we have structural features such as whether or not there is an envelope. The envelope refers to a lipid bilayer which can enclose the capsid of a virus. The envelope can also have peplomeres, which are extensions of glycoproteins. Glycoproteins refer to proteins formed from a polypeptide chain with a carbohydrate group attached and serve to assist the viron in entering a host cell. Another prominent variation in virus composition is the difference in the genome type present in the viron. So DNA and RNA are both sequential nucleotide chains and are able to code for structural proteins. In viruses, RNA and DNA can either be single-stranded, so this is ssDNA and ssRNA, or double-stranded, which is referred to as dsDNA and dsRNA. So RNA viruses can be further classified by their sense if they are single-stranded. The sense refers to the polarity of the nucleotides on the molecule, with positive ssRNA being in the correct order for immediate translation, and it doesn't require transcription to begin replication. Meanwhile, negative ssRNA is complementary to positive ssRNA. So must be transcribed first to create positive ssRNA. So the translation is just one stage in the life cycle of the virus. There's a few common elements that I'd like to run through. So these can be separated into stages belonging to either the intracellular phase or the extracellular phase. Starting from the extracellular phase with a fully intact viron, the virus uses a binding receptor to attach to a specific host cell. The next stage involves the internalization of the viral genome by one of the following methods: receptor-mediated endocytosis, fusion from without, and non-Clarin-mediated endocytosis. In the diagram here, you can see that the viron enters via endocytosis. So I'll give a bit more information on this one. This involves the virus binding to a specific virus receptor area and forming a coated pit from the protein Clarin, which lines the cellular membrane. From this, the cellular membrane is inverted to form an endosome inside the cytoplasm containing the virus. The virus degrades the integrity of the endosome through lowering the pH inside the endosome by releasing protons. This is kind of the most common observed method of internalization. Once a genome has been released into the cell's cytoplasm, it can either remain within the cytoplasm or enter the cell's nucleus. The next stage, the virus will begin the formation of viral mRNA via transcription, and following this synthesis, the host cell's ribosomes are commandeered for the expression of mRNA via translation into structural and non-structural proteins needed for the assembly of the virus. Non-structural proteins are involved in the virus's metabolic processes, but are not constituents of the physical viron. The penultimate stage is viron morphogenesis. These steps include the self-assembly, packing, and maturation. Self-assembly is a spontaneous process which involves the formation of an ordered nanostructure from a previously unordered system of subunits. This process can occur without any external input or force and tends towards equilibrium as it begins as a diffusive process. In relation to viruses, self-assembly is the basic mechanism responsible for the formation of the protein capsid containing the viral information. However, there is some variation in how this process occurs from RNA and DNA viruses. Simultaneously to self-assembly, the new viral genome is packed inside the capsid to form the nuclear capsid. Packing can trigger the final stage of viral morphogenesis, the maturation of the assembled virus to form a viron capable of infection. However, some varieties of virons do not undergo maturation until after being trafficked out of the cell. Finally, the life cycle is concluded with the releasing of the viron from the cell, so the viron re-enters the extracellular phase.
So, as Isabel said earlier, viruses are constructed by a genome strand surrounded by a protein shell known as a capsid. What I'm going to be talking about is the geometry of this capsid shell itself. But firstly, we need to understand that viruses want to be as efficient as possible when constructing this shell. It would therefore much rather have to encode a small number of proteins to construct the shell rather than a large number. Having a small number of proteins with having a small number of identical building blocks with specific bonding sites naturally creates a restriction on the number of possible geometric shapes a virus can construct. In fact, there are only two main shapes a virus can take, helical or icosahedral. Almost all viruses fall into one of these two categories, although a small number do not, but often the shapes these small numbers take have very similar properties to these two structures. So let's start by describing a helical virus. So if we look at the top left image, we can see that the backbone of a helical virus is the coiled genome strand at its center. We then have closely packed identical proteins between the turns of the coil. This pattern repeats the entire length of the genome strand, resulting in a long, flexible, rod-like structure, as can be seen in figures A and B, which are two electron microscope images of a tobacco mosaic virus and an Ebola virus. We tend to describe these helical structures with two parameters, the amplitude, which is the diameter of the helix itself, and the pitch, which is the displacement after one full turn. The discovery of these helical structures is quite interesting in its own right. It was first discovered by Rosalind Franklin, the same person who discovered the double helical structure of DNA, whilst experimenting with X-ray diffraction on a tobacco mosaic virus. And you can actually see her results in the top right image. She theorized that the horizontal maximums were a result of the coiled structure of a tobacco mosaic virus, and this was the first real attempt to describe the geometry of a virus. So we'll now move on to icosahedral viruses, which are more interesting viruses from a mathematical perspective. Icosahedral viruses are constructed by tiling proteins to create an almost spherical shaped capsid. In the case of a helical virus, we said that all the proteins were identical. If we follow this same assumption for the icosahedral virus, we end up with a very tight restriction on the number of unique structures that we can produce. Therefore, icosahedral viruses are not constructed by one set of identical proteins, but by multiple sets. This still offers a large amount of restriction in the geometry, but it offers enough freedom that we end up with a variety of different viruses. But now that we have a more elaborate structure, we need a good way of describing their complexity. To do this, we first embed a hexagonal lattice into the surface of the icosahedron, as can be seen in the top image. This hexagonal lattice can be used to describe the position of all the proteins on the surface of the capsid. From this lattice, we could get a value known as the triangulation number, which describes the complexity of the tiling pattern and is given by the equation on the slide, where H and K are the axes given in the image. What's quite important to note about these structures is their symmetry axes. So they have 2, 3, and 5-fold rotational symmetry axes. The high symmetry of these structures means that when a virus starts to undergo spontaneous self-assembly, you can start from any protein and it will always construct the exact same structure. So I just got a wee bit of experimental evidence to finish off this section. So following from Rosalind Franklin's death in 1958, a man called Aaron Klug took over research at King's College London, and he decided to repeat the X-ray diffraction experiment performed by Rosalind Franklin, but this time on a polio virus. What he got was a result in the left image, which had a very different diffraction pattern to the previous X-ray diffraction experiment done by Rosalind Franklin. What we can see here is point-like maximums extending radially outwards. And if we compare this to the expected diffraction pattern for an icosahedron, we can see that they're very similar with the lines corresponding to the symmetry axes in the structure. This was the first real piece of evidence for icosahedral structures in viruses. Both this and Klug's tiling theory, which was the theory we discussed in the previous slide, resulted in him actually getting the Nobel Prize for Chemistry for his work towards virology. And these other two examples I have up here, which I'm not going to try to pronounce their names, it's just too difficult. But I thought they were quite interesting. Image A shows a very good example of an enveloped virus, which is something that Isabel said in the previous section. And image B is an electron microscope image of a virus, and you can actually see, although it's kind of difficult, the triangular sides and the actual icosahedral shape of the virus. And that's all you will need to know about the geometry for this presentation. So with that, I will now pass you on to Alvaro and James, who will be talking a wee bit more about the thermodynamics of a virus.
So virus self-assembly is a spontaneous process. And we know from thermodynamics that for any spontaneous process to take place, there must be a decrease in the free energy. The equation for free energy has two components, an energetic and an entropic term, as you can see on the slide. When a virus capsid is assembled, the entropy of the constituents decreases as the capsid is in a highly ordered state compared to the previous state. This by itself results in an increase in free energy. So we know from this there must be more factors at play for the process to take place spontaneously. Primarily, there must be a decrease in the energy, which in this presentation we talk about as an increase in the internal energy. This must be some sort of binding energy. The main form of this energy is electrostatic, with a number of different contributing factors. There are also a number of contributing factors affecting the entropy, other than just the capsid. Whenever you're considering entropy, it's always important to consider the total entropy of the system, remembering the surroundings. So in this case, the entropy of the ions in the water must be considered. Alvaro is now going to take you through the different contributions.
As James said, the electrostatic interactions are the main contribution to internal energy. This is due to two reasons. First, both DNA and RNA are negatively charged molecules. And second, even though viral proteins might not have a net charge as a whole, they're usually positively charged at the area in contact with the genetic material. Thus, there is an attractive force between the genetic material and capsid subunits, which promotes assembly. And at the same time, there is a repulsive protein-protein interaction, which has the opposite effect. And the presence of ions affects significantly the electrostatic contribution. To illustrate this, let's see the diagram on the right. And let's imagine we have a positively charged capsid that is empty, so no genetic material inside, represented by this blue circle. And this situation can actually happen in nature because for some viruses, like most bacteriophages, the assembly process starts with the assembly of an empty capsid and proceeds with the injection of genetic material inside. So the empty capsid generates an electric field that attracts negative ions and repels positive ones. And in the end, the ions arrange in such a way that they reduce the strength of the capsid electric field, producing what is known as the screening effect. This effect can also happen with genetic material inside the capsid. For instance, in many ssRNA viruses, such as the Brom mosaic virus, the capsid will assemble around the genetic material. And the result is that the number of negative charges on the genome significantly exceeds the number of positive charges on capsid proteins, having an overall negative charge. In those cases, positive ions are attracted and negative ions are repelled, and nonetheless the same screening effect is produced. As mentioned earlier, upon assembly, the entropy of the viral components obviously decreases. This is also the case for the ions entropy, since the ions are arranged in order to screen the capsid or virus electric field. And these two entropic contributions increase the free energy and therefore promote the disassembled state. However, there is a third entropic contribution coming from the medium, in other words, the water, which arises from the so-called hydrophobic interactions. These interactions are a consequence of the polar nature of water molecules. Water molecules behave as dipoles, forming arrays of hydrogen bonds, as shown in the diagram. And well, there are many ways in which this can be done with the regular structure of molecules, if they are not blocked or interrupted by an apolar molecule. So let's imagine we introduce an apolar molecule here. Then certain hydrogen bonds would be impossible to form. And viral proteins often have apolar patches that are hydrophobic, repelling the water molecules. So by forming clusters of proteins, the apolar surface exposed to water molecules is decreased. And this has a small electrostatic effect, but more importantly, it increases the number of configurations that water can take. So overall, there is an increase in entropy, and thus these hydrophobic interactions promote the capsid assembly. And another relevant driving force in virus self-assembly is the so-called bridging interaction. This happens when genetic material, which can be thought of as a chain made of charges known as monomers, represented in the diagram here as a red chain, adheres to protein tails, which are these small yellow tails, of different capsid subunits, which are these blue pentagons made of proteins, which are represented by blue spheres. So the capsid subunits repel each other as they have the same charge. However, it is entropically favorable for the genome chain to be in an unextended entangled state, pretty much like my earphones every time I leave them inside my backpack. And this contraction of the chain overcomes the electrostatic repulsion and results in the assembly of the virus, as we can see in the diagram.
We've now seen the different interactions, but the environment the capsid is in must also be carefully considered. Each of the factors mentioned are carefully balanced, and their energy will depend on the environmental factors such as pH and concentration, as well as temperature. This will result in certain conditions favouring assembly or disassembly. This is important as once a virus has entered a host cell, it must disassemble to deliver the genetic material. Temperature is the simplest, as we can see it in the free energy equation, effectively making energy more important at low temperatures and entropy more important at higher temperatures. But remember, the temperature range of most viruses is quite small, so this isn't the most important factor. The pH must be considered both inside and outside the capsid. pH has an effect because a change in pH changes the charge distribution of the proteins, then affecting the energy in the interactions. An increase in either internal or external pH reduces the free energy, making assembly more likely. However, the external pH, as you can see in the graph, has been experimentally shown to have a much greater effect, reducing the energy by 2 orders of magnitude more than a change in internal pH would. The concentration of ions and of proteins needs to be considered as well. An increased concentration of ions increases the free energy of the initial state, making the intermediate state a more energetically favorable state. This is known as assembly active conformation. The concentration of the proteins has an entropic effect. At high concentrations of proteins, the entropic increase that arises from the formation of the capsid is smaller. As it's an entropic term, there is a logarithmic relation between the free energy and the protein concentration, as you can see in the top graph. This also has an effect on the kinetics. There are 2 different kinetic traps that must be thought about. If the environmental conditions result in very fast nucleation compared to capsid growth, there may be very few full capsids completed as the concentration of proteins decreases too quickly. The second trap occurs when the protein interaction is too strong. In this case, the interaction strength results in interactions being energetically favorable that don't actually result in full capsid growth.
So quite often, the goal for physical virologists is to derive an expression for the free energy of the self-assembly process, which ideally depends on variables that are related to external factors such as temperature or pH. And to derive these kinds of expressions, electromagnetism and statistical mechanics techniques can be used to model both icosahedral and helical viruses. For instance, Antonio Sieber and co-workers considered a model of the assembly of a protein capsid around an RNA molecule in which the ions were treated as an ideal gas. This is known as the Poisson-Boltzmann approach. Moreover, they derived an expression that related the free energy with ionic concentration and optimal genome length. The results are shown in the graph at the right, and where we can see the free energy plotted against the number of monomers, N, in the genome chain for different ionic concentrations. By locating the minimum of the free energy, we can estimate the optimal genome length, because we can get the optimal number of monomers in the chain, which is equivalent. These models provide a fundamental understanding of the underpinning physics that explains why certain environmental conditions promote virus self-assembly. And they can also explain why certain virus features, such as the capsid geometry or certain values for the genome length, are found more often in nature. So we will continue with the mechanical properties of viruses. And this will be the turn of Ewan.
As mentioned before, I'm now going to talk for a bit about mechanical virology, which is the study of the mechanical and material properties of viruses. An obvious question to ask is, why do we care about the material and mechanical properties of viruses? Well, to start with, viruses have unique mechanical properties that are of interest to material science and soft matter. They are both sufficiently strong enough to withstand the molecular turbulence of conditions in the body and the host cell, but have to be weak enough to be able to disassemble and release their genetic material when required. Additionally, a rapidly expanding field of research is the study of virus-like particles in the context of nanotechnology. Viruses are being used in applications such as targeted drug delivery systems. If you're designing a virus-like nanocage to deliver a drug to a particular tumor or organ, you will require that that virus be mechanically strong enough to reach its target location, and you will require it to release its cargo when required. Understanding the mechanical properties of viruses is of great importance to designing such systems. The primary method for measuring the mechanical properties of viruses is atomic force microscopy. Effectively, the atomic force microscope is used as a force spectrometer. It applies a force to the virus and measures its mechanical response. It does this using a fine tip of a probe to indent the virus. The virus then exerts a force back on the probe, which causes a deflection in the cantilever. We can then plot a force displacement graph, and from the linear region, determine an effective spring constant. In effect, what we have here is a composite spring system made up of the virus and the cantilever. The spring constants add in reciprocal, and we can rearrange to find an equation as shown on the slides. The issue here is that the spring constant is not an intrinsic material property, so its value as a comparative tool is somewhat limited. Viruses, as we've seen, have different geometry and shapes and sizes. Comparing a large helical virus with a smaller icosahedral virus will not be useful when using the spring constant. Instead, we can use the intrinsic elasticity as quantified by the Young's modulus. Unfortunately, we cannot determine this directly and must use a set of approximations treating the virus as a spherical, homogeneous, empty shell. We can use these approximations to determine an expression that relates to spring constant and the Young's modulus, as shown on the slides. Here, E is the Young's modulus, H is the thickness of the shell, R the radius, and alpha a dimensionless constant, which is usually around unity. We have assumed that H is sufficiently smaller than R to give the limiting form as shown on the slides. Armed with these tools, we can investigate the effects of different factors on the mechanical properties of viruses. Firstly, we can compare what effect the genome has on overall virus stability. As Alvaro previously mentioned, there is an attractive electrostatic interaction between the capsid proteins and the genome. Hence, the genome is expected to have a stabilizing effect. This is observed. In general, we note the following trend, that viruses that self-assemble empty and insert the genome after, such as a bacteriophage, using a packaging motor, are in general mechanically stronger than viruses that self-assemble around the genome. This is due to the greater mechanical pressure associated with the former method of self-assembly. Additionally, some viruses, such as bacteriophages, mature over the course of their life cycle. For bacteriophages, maturation causes a reorganization of the protein subunits and results in the virus becoming mechanically stronger, which is required for the life cycle of the bacteriophage, which utilizes a packaging motor. However, for HIV, the inverse is observed. Immature HIV capsids are significantly more robust than mature capsids. This is also observed for MLV. It is predicted that the reason for immature HIV capsids being more mechanically robust is linked to the infectious nature of viruses themselves. It has been found that immature HIV viruses are unable to effectively enter cells because they are unable to fuse effectively with the cell membrane. It is speculated that this leads to a regulation. This prevents the immature viruses from entering cells and is important for efficient infection. This establishes a direct link between the mechanical properties of viruses and the infection process. Also, environmental factors have been seen to be important. As we heard from James, pH can have an impact on capsid self-assembly and stability. Norovirus has been found to be mechanically stronger under acidic conditions, such as in the gastrointestinal tract and stomach, where infection is not supposed to take place and the virus is meant to travel unimpacted. However, it's also been found to be weaker under alkaline conditions, such as that in the ileum, where infection is supposed to take place. Now we turn our attention towards bioengineering. Designing engineered nanoparticles for drug delivery must take into account the factors we have discussed. For example, if you want to deliver a drug to the pancreas, you will require a virus that is able to travel there uninterrupted and will be able to reach its material there as required. If the pancreas is acidic or alkaline, you'll need to take that into account when designing this nanoparticle. Likewise, for other applications, such as vaccines or contrast agents, the virus will need to be mechanically strong enough to serve its purpose. In the case of vaccines, the capsid will have to remain intact long enough to induce the required immune response. I'll now hand over to Niamh to discuss some of these applications in more detail.
The physical properties of viruses comprise of unique and incredibly useful attributes. Over the next few slides, I'll mention a few areas of study today that are exploiting these properties in order to improve people's lives. Some of the most prominent areas of research are being done in the medical field. Virus capsids are being developed for a number of medical procedures and treatments, which I will mention next, along with developments in renewable energy. The picture on the left shows a tobacco mosaic virus genetically engineered to express a cysteine residue, which is shown on the edge of the molecule in yellow. This image was taken from a paper from Royston et al. In 2008, which used the protein expression to bind the virus to other materials such as gold. The self-assembly of the TMV virus created an ordered template for the synthesis of other materials which could bind and grow. This was used in batteries to prolong the life of the electrodes. These applications have promising futures in the biotechnology fields. However, here I will choose to focus on the medical applications and renewable energy. Following on from Ewan, the delivery of drugs into the human body and to the area of importance is crucial in the effectiveness of the drug and the well-being of the patient. For cases such as cancerous tumours, targeting the drug to deposit at the tumour site is crucial. A common chemotherapy drug, doxorubicin, or DOX, is hardly cardiotoxic. This limits the use of the drug in patients, but developments in using self-assembling viral capsids as carriers has promising results in reducing the toxicity while still remaining effective. As mentioned previously, self-assembling capsids are highly symmetric and shown in the figure of the cucumber mosaic virus on the right. The 2, 3, and 5-fold axes are annotated. A unique feature of this virus is the quasi-3 and 6-fold symmetry axis, with the quasi-6-fold axis adopting small holes. These gaps in structure allow the intake and distribution of docks through electrostatic interactions, which was explained by Alvaro. The positively charged docks and negatively charged genome in the center of the virus attract each other. Another important aspect of this delivery system is the target. Tumors can be described as an unorganized jumble of cells, which allows a property called the enhanced permeability and retention effect, or the EPR effect, to be employed. Enhanced permeability means fluids are not drained from the tumor, and therefore the docks or any other substance of similar size to viruses retain for longer, allowing a large distribution of drug to the site. In other instances, the outer capsid can be manipulated, where specific targeting molecules can be incorporated to the outer protein, which in turn can bind to certain target sites. On the topic of delivering specific material into the body, the same process can be beneficial in the improvement of MRI and PET scans. In the image at the top of the screen, a gadolinium ion complex is seen to be attached to the outer and interior capsid. This ion is commonly used to enhance the relaxation time of proton spins when in a magnetic field applied to the body. The EPR effect also prevails here, with the capsids congregating in tumors or specific troubled tissue, and causes a large contrast in colors for imaging purposes and detection of the affected tissue. Similarly, PET scans use the same method but load a radioactive element which increases the amount of positron annihilation and signals detected in the infected tissue. An animation of the PET process is shown at the bottom of the slide. On a different note, self-assembling viruses can be manipulated to express fluorescent proteins, which when excited by light can follow a Foster energy process. This requires two particles to be in close proximity, and this excitement from light is shown by the purple arrow. When two viruses assemble at the same time, an energy transfer can occur between them. This process is shown with the blue arrow in the figure to the right. This energy can then be converted into hydrogen by carbon nanotubes, which can then be utilized. It is hoped this process can be used in solar cells and help in the sustainability development of energy. Understanding the physical properties of viruses has led to incredible advancements in many fields. Without knowledge of structure, thermodynamic properties, and areas such as mechanical manipulation, developments in cancer drugs and sustainable energy that have the possibility to save and improve lives would not be able to happen. Thank you very much for listening.